We have read that most of the free energy that is released during the oxidation of glucose to carbon dioxide in glycolysis and TCA cycle is retained in the reduced coenzymes NADH and FADH2. Oxidative phosphorylation is the process in which this free energy is released by the oxidation of NADH and FADH2 and is used in the formation of ATP molecule. Since here ATP is produced due to oxidation process, it is known as oxidative phosphorylation. Unlike substrate level phosphorylation that we have seen in glycolysis and TCA cycle. Electrons that are released due to oxidation are transferred to oxygen and water molecule is formed. From NADH and FADH2, electrons are transferred to oxygen through a series of electron carriers present on the inner mitochondrial membrane. These electron carriers are grouped into 4 large respiratory enzyme complexes, each containing transmembrane proteins that hold the complex. These 4 major respiratory enzyme complex of the electron transport chain in the inner mitochondrial membrane are complex 1 NADH coenzyme Q reductase, also known as NADH dehydrogenase, complex 2 succinate coenzyme Q reductase, complex 3 coenzyme Q cytochrome C reductase, also known as cytochrome BC1 complex, and complex 4 cytochrome C oxidase. These enzyme complexes are multimeric in structure and comprise prosthetic groups such as flavins, heme, iron sulfur clusters, and copper. Electrons in the electron transport chain start with a very high energy and gradually lose it as they pass along the chain. They are transferred by the reversible reduction and oxidation of iron sulfur clusters, coenzyme Q, cytochromes, and copper ions. Each carrier in the electron transport chain accepts an electron or an electron pair from a carrier with a less positive reduction potential and transfers the electron to a carrier with a more positive reduction potential. Thus, such reduction potentials of the electron carriers in the electron transport chain favors unidirectional electron flow from NADH FADH2 to oxygen. Also, there is a fifth protein complex in the inner mitochondrial membrane known as ATP synthase. It catalyzes the synthesis of ATP from ADP and inorganic phosphate. ATP synthase is driven by a flux of protons across a gradient which is generated by the electron transfer in the electron transport chain.